Hello, everyone. We will begin within the next minute or so. We just want everybody to log on. We have a pretty large list today, so we'll just give it a few minutes. Thank you.
את יכולה להצטרף. מצטרפת? את יכולה להצטרף פה. אני לא שמתי את עצמי על מיוט.
Good morning, good afternoon, and good evening from wherever you're joining us today. My name is Sharon Frankel. I'm the Executive Director of the Canadian Friends of Tel Aviv University in Montreal. Over the past 2 months, together with our colleagues in Toronto, Tel Aviv University and its friends associations around the globe, we've been offering webinars as a way of staying connected and help keep our minds stimulated and entertained during these challenging times. We've held some excellent webinars, and there are some more amazing in the pipeline, and today will be no exception. As the pandemic forced people around the world to slow down, Tel Aviv University literally dropped everything to turn its focus on combating the coronavirus. It all started by leading Tau professors at the forefront of some major advances, dropping everything and working with their teams, now comprised of some 120 researchers, 24/7 to find a treatment, a cure, and a vaccine. And their efforts have started to pay off, as you will hear today from Professor Jonathan Gershone, who I personally thank for having agreed to a second special webinar hosted by us in partnership with the Spanish and Portuguese Synagogue. And thank you to everyone at the Spanish for preparing and working with us on this webinar. Mid-May, we launched the Drop Everything Urgent COVID-19 Appeal, the university's first ever international large-scale crowdfunding campaign to support Professor Gershone and his colleagues. The construction of a COVID-19 national test center on campus that was literally built.
Sharon, we can't hear you. Sharon, we can't hear you well. Try again.
My apologies. As well as assist about 6,000 students who are relying on their university and its donors to support them financially, emotionally, and psychologically during this crisis. We are proud of the leading role Tel Aviv University is playing, finding solutions to these challenges, as well as the global battle we are all currently facing. Our $2 million campaign, matched dollar by dollar, thanks to some international donors, and thus doubling each gift we receive, is enabling Tel Aviv University to do all it can to fight and understand the horrors of this global pandemic. And just like Professor Gershone is 2/3 of the way in his work, so are we with this campaign with just over $1.4 million. Please, following today's webinar, drop everything. Let's continue to fight COVID-19 together, and please help us to reach our campaign goal by June 30th. You can donate to our campaign at telavivuniversity.crowdchange.net. I will be posting the website again in the chat box throughout this webinar. Your generous support will help ensure that the extraordinary work being done on campus can and will continue. And now, it is my pleasure to ask Tel Aviv University Vice President Amos Ellard to say a few words. Amos, thank you very much for joining us today.
Thank you, everyone. Shalom. Thank you, Sharon. It's my privilege to be here today in the name of the Tel Aviv University, and really with this esteemed panel and really guests with us. In the name of Tel Aviv University, I know this is a strange situation where we are doing those webinars, those calls online, because the world has been changed, and we're going to hear from some esteemed people to tell us what they're doing about it today. But I really want to force what Sharon said. Tel Aviv University from day one really took it very seriously. From over 180 laboratories that are working in humanities, social sciences, and of course exact sciences on coronavirus, and from establishing now the pandemic center, really trying to force for the future of the world how to solve pandemic issues. Of course, right now the coronavirus, the COVID-19, which Professor Gershone is one of the brightest one that is working on, but also on other issues for the future of Israel and of course the world. Tel Aviv University is also doing a lot on other areas, of course, as you know, and we're also helping a lot of students in the university that are suffering from this situation. It's funny how everyone is talking about online services, and there's big plans of how to move from academic world into online and what's the future for that. And exactly in a week and a half, the whole university, I'm talking about 30,000 students, really went online. All courses today in the university are given online. And of course, we are hoping to be able to open in October, but we don't know yet. But that's the plans for now. International students are still planning to come, but again, maybe Professor Gershone can tell us what the future will hold. But we, of course, in Israel and in the university are optimistic. We want to thank everyone, and we want to pray for everyone's safety and everyone's health. It's a special event that everyone is unified in this terrible predicament, really this situation. And I really want to thank the leadership of Tel Aviv University in Canada and the Canadian Friends of Tel Aviv University, really helping the university in this time and helping and really bringing us, all of us together. So Sharon, thank you again, especially to you for running this special campaign, and I hope the strong message that we'll hear today from Professor Gershone and the team will help and continue the efforts. Thank you very much, everyone. Thank you. Toda rabba.
Thank you so much, Amos. Thank you, Sharon. Shalom to all and bruchim habayim. It's an honor for the Spanish and Portuguese congregation to partnership with the university, Tel Aviv University, such a pertinent presentation. It's a special treat for me as a born and raised Israeli until my parents' sudden migration to Canada in the late 1980s. I want to thank the entire Spanish and Portuguese membership and all of you that are here today. A special thanks to our dearest friend and member, Mrs. Gladys Katan, for this great initiative. I thank Professor Gershone and the University of Tel Aviv for taking the time to share with us the latest developments in regards to the COVID-19 vaccine, a great concern for the worldwide community. I'm sure you all have a lot of questions. So without any further delay, I would like to welcome the Consul General of Israel for Canada and the Atlantic provinces, Mr. David Levy. I remember still today the encouraging and inspiring words he shared with our community on his last visit to our synagogue. Mr. Levy more recently served as a political counselor at the UN and international organization, human rights departments. But more importantly, a triple graduate from the Tel Aviv University. It's an honor to have him with us. Please welcome Mr. David Levy. Thank you so much.
Shalom. ערב טוב לכל המשתתפים בישראל. Bonjour tout le monde à Montréal. Merci d'être avec nous aujourd'hui. And thank you to everybody across Canada for being with us today. I'm very honored to participate in this webinar, both as Consul General of Israel to Quebec and the Atlantic provinces, but also, of course, as a proud alumnus of Tel Aviv University. I even wore my EAU pin, especially today. I'm very excited to introduce today's special guest, Tel Aviv University Professor Jonathan Gershone. With your permission, I'll go briefly over his very impressive bio. Born in Israel and completed his bachelor's in biology and PhD in biochemistry at the Hebrew University of Jerusalem. Later did his postdoctoral training at the Yale School of Medicine. Returned to Israel in 1983 and joined the Department of Biophysics at the Weizmann Institute of Science. In 1988, he joined Dr. Robert Gallo at the Laboratory of Tumor Cell Biology at the National Institute of Health in Bethesda to work on AIDS therapy and prevention. In 1990, he returned to Israel to the newly established Department of Cell Research and Immunology at Tel Aviv University. Over the last decades, he has focused on developing methods for the rational design of vaccines to such pandemic diseases as AIDS, hepatitis C, influenza, and SARS. Professor Gershone was recently awarded a US patent for his innovative vaccine design for the corona family of viruses. And he has partnered just last month with Swiss pharma company Neovi to fast track his development of the COVID-19 vaccine. Professor Gershone has escalated the work in his lab and is now 2/3 of the way of developing a vaccine candidate against COVID-19. He lives in Israel with his wife and has 3 daughters and 10 grandchildren. On a slightly personal note, when I go to the Spanish and Portuguese or to other synagogues in Montreal, people approach me and ask me, do you know my cousin, Yuda? You know my friend, Shmuel? As if Israel is a very small place. Everybody knows everybody. Well, guess what? For many years, the Gershone family lived in Rehovot, a couple of blocks from where I grew. And the reason I know that is because I went to school with Professor Gershone's daughter, Roni, and we're close friends and we keep in touch until today. Of course, at the time, I didn't know him as Professor Gershone. For me, he was Johnny, the guitar player, but I'm very excited and I'm very eager to hear how the vaccine development is progressing and what are his thoughts about the future of this pandemic. So thank you all for being here today, and I hope to meet you all in person soon.
Gladys, you're on.
Hello, I'm the governor of Tel Aviv University, as well as member of the board of Canadian Friends of Tel Aviv University. I wish to thank our membership of the Spanish and Portuguese for joining us today to hear what Professor Gershone has to say. I'd like to thank everybody who Rabbi Pinto, Amos, Ellard, Sharon, Consul General David Levy, and special thanks to Professor Gershone for taking the time to be with us and to answer our questions. So without further ado, I'm gonna start with the questions. What we'd really like to understand is how these vaccines that you're working on and that others are working on all over the world really work. And in order to understand that, I think the basic question is, how is a virus different from bacteria? And how is different than the flu virus?
Okay.
Professor
Well,
Gershone.
Yep. Well, thank you very, very much. I also want to thank those that introduced me. David, thank you. Certainly a surprise. And yes, I continue to play the guitar as poorly as I've always, but I continue to enjoy it. So thank you. With regard to understanding vaccines, which I hope to be able to develop during the course of this webinar, I agree that it first is good to maybe just set the scene and try to clarify some distinctions between viruses and bacteria, because they're very different and the approach has to be therefore very different. Generally speaking, living cells store the information in DNA. And in fact, it's become also a part of our current language. We all say, well, you know, it's in my DNA, or preferably it's not in my DNA. And so DNA is the molecule that sits in our nucleus and stores the information, the blueprint for everything we do in life. And then subsequently, the DNA, a copy of it is made into RNA, another molecule, which is ultimately translated into proteins. So I don't want to get into all the chemistry and so on, but basically you have to know that information flow in living cells goes from our genes, our DNA, through an intermediate copy, a Xerox copy of our DNA, RNA, and protein. And why is this important? All living cells have DNA, RNA, and protein, 3 components. And bacteria, which are very, very small cells, have DNA, RNA, and protein, because basically they're alive. Now, in marked distinction to bacteria that can cause disease, but as living organisms, viruses, surprisingly, are not alive. They're particles. And why do I categorically make that distinction? It's because viruses contain either DNA, so all viruses in the world can either be DNA viruses, or they have RNA, and they can have RNA viruses. And so there are no viruses that have DNA, RNA, and they can't make protein. So viruses, under the circumstances, are very different from bacteria in the fact that they cannot replicate on their own. They do not have the capacity to convert the information in the virus into protein, into living organisms. And for all of us who may not necessarily be biology savvy, you have to think of a virus like a disk on key, like a key drive. And we all know that disks on keys, they're not computers. However, they contain information. They're coated with the plastic encasing, and there's a chip inside. And the information in a disk on key becomes viable and effective if you can plug it into your computer in the USB port. And so viruses are very similar. They contain information, either in the form of DNA or RNA, but never both. And they can only cause damage and disease once they've been plugged into the cells of our body. And so they do this through a process they have on the body's cells, something that is somewhat a unique USB port of the virus. It's a receptor. It's a protein, which part of the virus binds and hooks into, and only when that physical connection between the port on the one hand, the receptor on the cell, and the protein of the virus interact and bind, is there an infectious process. And the information of the virus can be transported into cell and then cause disease. Now, because viruses are very different from bacteria that are living organisms, the medicines and the treatments for bacterial diseases are not effective at all on viruses. And antibiotics, which we all know how important penicillin and tetracycline and other antibiotics have had on the impact of human health, viruses are not susceptible to antibiotics. And in fact, they pose a very difficult problem in trying to resolve diseases caused by viruses because antibiotics don't work. And in fact, viruses use all of the cell machinery of our body to replicate. So anything that we discover that may interfere with the replication cycle of a virus could very likely also poison us, the host. And so therefore, it's a very fine line between finding materials that can knock out viruses and on the other hand, leave us, the patients, safe and healthy. So we now know that viruses aren't living organisms, they're particles. They can either be DNA viruses or RNA viruses. And to go one step further, we now also will learn that the COVID-19 virus, SARS coronavirus 2, is in the class of RNA viruses. And that's very important because RNA viruses have traditionally been difficult to treat. We know a number of examples of RNA viruses, such as HIV/AIDS. It's an RNA virus. And I don't know if you were familiar with Mark Weinberg, who was from Montreal. So he was Canadian and unfortunately passed away about 2 years ago. But Mark was very, very important and a senior pioneer in AIDS research and a very dear friend of mine and a dear friend of Israel. And so I take the opportunity, amongst others, to give kavod to Canada and to Canadian scientists, because a lot of what we do stems for our ability to network, interact, exchange, and collaborate. And so this is very important as well. We can't do it alone. We obviously need your help as far as donations, but that's another field that I don't deal with. I just deal with the science. But I have collaborated with dear friends in Montreal and in Toronto currently. And we're all working to try to figure out how to deal with the unique characteristics of viruses. And coronavirus is an RNA virus, and it is one of 7 coronaviruses that cause disease in humans. And we can continue further, but Gladys, I would be happy to take another question.
Okay, last time it was in May when you gave your speech and you talked that you were 2/3 away from developing a vaccine and a few months away from completing it. Are we advanced now? How far are we now?
About 2/3 in a month less.
Professor Gershone,
I'm obviously kidding.
So we haven't moved further.
The process of developing a vaccine is highly complicated. And I'll try to elaborate a bit. And people have asked me, is there light at the end of the tunnel? And the definitive answer is yes. However, the tunnel's very long. And so it's going to take us some time to get to that light. The reality is that we have been studying coronaviruses specifically since 2004. So whereas we previously heard that we stopped and dropped everything, most of the labs at Tel Aviv University who are working aggressively and enthusiastically in trying to solve the COVID-19 epidemic may have been working in various fields and dropped everything to now focus on SARS coronavirus 2. We have been working on SARS corona since 2004 and trying to figure out what would be a good candidate for a SARS vaccine. And I can elaborate that in a minute, but to your question, after working on SARS coronavirus, we continued our work on MERS in 2012. And so it took us 11 years from 2004 to 2015 to get to the point where we realized that we've actually developed a general solution to coronavirus vaccine development. And that is the reason why in 2015, we filed for patent. People, for whatever reason, believed that because there was the emergence of COVID-19, we sat down, wrote a patent, submitted it, and it was approved. Actually, it took us 5 years of prosecution from 2015 to get approval. And so it's very, very fortuitous. Now, the moment the Chinese published the RNA, the gene structure of the SARS coronavirus, which certainly is to the credit of the Chinese. People are arguing that they've been late and they've been devious and possibly withheld information. The reality is January 9, they published the complete genetic structure of this new virus, which is a remarkable achievement. Once we saw that in January, we began to plan and design the SARS coronavirus 2 vaccine as we view it. And this takes time. And when we did the same for SARS in 2002 and MERS in 2012, it took us about 4 years to translate our ideas into viable vaccine candidates. And here we started working actually in February. And in May, I said we have about another month or two. And I would say that we're pretty much staying on track. It's a very difficult task to condense 4 years of work into only 4 or 5 months. But actually, the experience that we gained over 15 years of research on coronavirus makes it feasible. And so I do believe that in the course of the next month or two at max, we should have the prototype of the kind of vaccine that we would like to promote. However, and being a scientist, I tend to be precise. We have to understand the difference between being able to say, here we have succeeded, we have the prototype. That does not mean that we have a vaccine in hand and that very shortly the public will be able to benefit from it. And this will take yet at least another year. And I believe that we are no later than most of the other vaccine modalities that people are trying to develop. We hear about the vaccine by Oxford University, the Chadox vaccine. We hear about Johnson & Johnson and their vaccine. And of course, quite a lot about the Moderna RNA vaccine. These are all excellent candidates. They're all the products of enormous efforts of very talented and gifted scientists. However, one has to realize that before a vaccine can actually reach the public and do benefit, it has to undergo preclinical trials and then phase 1, phase 2, phase 3 clinical trials. This takes time. And there are various aspects in the development of a vaccine that you can't speed up, neither with money or with energy. Sometimes we have to follow processes which take time. And so in my case, we're making very significant progress. I do believe that we'll have a prototype in the coming month or so. And then from there, we have already partnered with the Swiss company Aviv that will help us take it from the prototype and the design to a bona fide testable vaccine to be able to push through clinical trials and hopefully reach the public.
Thank you. We have the month of July for a prototype, approximately, and another year for all the trials to take place. I have questions about, you said you patented your research, your prototype, you have a patent for it. Are any of these other companies, Johnson & Johnson, Moderna, and Oxford, using your patent research?
The easy answer is no. the question that I would like to propose is that they will ultimately realize that they should. And the reason is the following. We've all had extensive experience in being vaccinated. And we all know, even if we don't fully understand the mechanisms of vaccines, we understand that it's usually not a single shot that we receive. And we typically receive our initial vaccination, which is then followed by boosts. And the reason is that a vaccine is like boot camp for our immune system. It's a training system. It allows our body and our immune system to scrutinize and analyze and try to better understand the structure of the virus in this case, and begin to learn what would be the best mechanism to knock it out. Can we find vulnerable aspects of the virus? Can we find its Achilles heel? What works best to actually knock out the virus? So this is a process of learning and trial and error, and this takes time. And so when you become naturally infected, it's a battle amongst the viruses on the one hand and our body on the other, and the effort to learn and knock out the virus. But very often the virus gets the upper hand and we're ill and sometimes die. On the other hand, if we can train the immune system in advance, and this is what a vaccine does, we can learn and study about the virus. We can develop the tools to knock out the virus under circumstances and conditions that don't cause illness, because the vaccine trains the immune system without causing disease. So that's the beautiful aspect of vaccines. Now, this takes time, and it sometimes requires, and most often requires, a number of rounds of training. Practice, 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 if you will. And so the first injection gives us the ability to begin to understand how to knock out the virus. But then in order to get really a very robust and effective vaccine in place, we have to undergo boosts, and sometimes 2 or 3 boosts. And so I believe that whoever is able to put out a vaccine in its first generation is going to require a boost. And I think that our particular technology is not only suitable to be a primary vaccine, but it is especially effective as a boost. And the reason is that our vaccine, in contrast to all the others, targets the immune system to the weakest spot, the soft spot, the Achilles heel of the virus. And the other vaccines do not. And it turns out that in some systems, in some viral infections, not focusing and generating an immune response to every aspect of the virus is sometimes not very efficient, but can also be dangerous. And in order to reduce the danger of generating useless antibodies, useless responses that could exacerbate and enhance the disease, a boost like ours would be especially attuned to that. And thus far, Johnson & Johnson, Moderna, Oxford, and others don't have the technology nor the ability to generate a vaccine that we've been developing for over 15 years. And so I think it's a very opportune situation where ultimately they will be able to promote their product and embrace ours as their effective next step boost. And so I'm encouraged, but of course, the first thing we have to do is deliver the goods. And so this is taking time, and we're developing it.
Thank you. There's a few questions that are popping up, and to do with especially, do we think that there is going to be a second wave, a bad one? Do we think that they will develop herd immunity, or are there any similarities with the SARS-CoV-2 with the original SARS-2 in 2003? So herd immunity, second wave, how does it compare?
I'm just jotting down. And okay, so as far as the second wave is concerned, this is obviously something that I have no idea. But in reality, I think that people have to seriously begin to understand that the immediate task is not to knock out and beat the virus, so to say. This is not going to happen. And so what we really need to learn is how to live with the virus, how to live with corona. And this, for the moment, in the absence of good therapeutics and vaccines, requires that we have to embrace, understand, and respect the preventive behavior that is most effective. And this is at a number of levels. First of all, we all recognize that the isolation, the quarantines have been extremely effective in dropping the rate of infection, and if you will, by flattening the curve, but it hasn't eradicated, it hasn't removed COVID-19 virus from our presence. In fact, there's been no change. The virus is with us. It hasn't changed. It's no worse. It's no better. And the only thing that really has changed is if initially the first case of COVID-19 was January 26th in Canada. And since then, there are about 90,000 people who've been infected. So if we consider this, late January, beginning of February, there were only very few people that could have spread the disease in Canada. And now there are at least 90,000 that have experienced the disease, and many of them are still contagious. And so we have to be very, very sensitive to the fact that the virus is with us. We have not removed it. But quarantine and isolation has been effective. Donning masks is very effective, provided that we all wear masks. Because those of us who are not infected, the mask is marginally effective. But those of us who are asymptomatic but contagious and spreading the disease, the mask is very effective in preventing the spread of micro droplets from sneezing and coughing and speaking and so on. So that's very important. And of course, hand washing and hygiene. Now, why do I say this regarding the second wave? If we lose sight of the very fundamental and easy tools we have in behavior, social distancing to some degree, we don't have to shut down. We need to revive the economy. We have to let people live, because this is a long-term effort. It's going to take at least another year, a year and a half before we have a vaccine. And so people are going to have to assume behavior which is going to be compatible with keeping the numbers down. So we have to wash, we have to wear masks, and so on. And if we can do that, then the chances of a very serious emergence are much reduced. And think about it. When you go into your car and you sit near the driver's seat, there are very, very few people today that would dare to turn on the ignition without first putting on a safety belt. And a seat belt has become common practice. Now, when I was a kid, there were no seat belts. And when my kids were kids, there were no seat belts in the back of the car, in the back seat. And it's only in the last generations that this has become totally natural, that when you get in the car, you put on your seatbelt. And this should be the same type of behavior that we have to embrace, we have to assume and practice safe behavior, because the virus is with us for the long term. Now, one of the things that people have mentioned is that maybe herd immunity will save us. And the problem with that is that herd immunity is extremely, extremely expensive to implement. What does this mean? Herd immunity is when the virus no longer finds anybody to infect in the immediate circles of people and so on. So in order for herd immunity to really be effective, it means that 80 to 90% of the population has to have either been vaccinated, and that's what we do with vaccines, or has experienced the disease. Think about that. If we're talking about 80, 90% efficacy, in Israel, we have 9 million people. Well, because let's say 1% of all those that get infected are going to die, in order to develop herd immunity, we're going to have to tolerate 80,000 deaths in Israel, which is totally and completely unacceptable. And then if you multiply those numbers in each and every country, Canada, the United States, these are huge, huge numbers. And the chances of reaching the level of protective immunity in the community are very, very, very slim. And so I think that academically, it's an interesting argument, and it's totally futile to promote, definitely not as a policy. And we see what's going now on in the UK, where Johnson had initially said, you know, we're not going to do anything, herd immunity will take care of us. And unfortunately, those who were advising him didn't explain completely the cost of trying to reach that level. Now, the third question I haven't forgotten was, how do we compare the two viruses, SARS coronavirus from 2002 and SARS coronavirus 2020? And the reason why we are asking that is because the lessons we've learned from SARS coronavirus in 2002 was actually a very encouraging situation. The pandemic initiated in Guangdong in China and spread to about 20 countries. It started in November of 2002, but lo and behold, by July 2003, 9 months later, the virus petered out. It simply disappeared. Now, was this a God-sent miracle? I won't ask the rabbi, but I know that God says that we have to also invest in our own and not let everything in his hands to deal with it. So actually, social distancing was implemented in 2002. A total of 8,000 people were infected. So today in the United States, some 20,000 people are infected daily. So we're looking at a very different beast. We're looking at a virus in 2002 that over 9 months infected 8,000 people. Unfortunately, at a cost of 10% fatality. So about 800 people died. However, the SARS coronavirus 2, that is from 2020, what we're dealing with now, has already infected well over 7 million people. More than 400,000 people have died in over 150 countries. And we're looking at a very, very different virus. First of all, it's already summer. And if you look at some of the countries that are now experiencing rampant, really serious infectious rates, such as in India and in Brazil and in Argentina and of course Mexico and in Israel for that matter, all of these countries are already hot. So the question that people assumed that the virus will peter out once we hit summer is just not going to happen. We already see that temperature per se is not going to kill the virus. Secondly, in SARS coronavirus from 2002, only the very ill, those that had clear clinical symptoms, that they had high temperature and they were sneezing, coughing, they didn't look good, these people were the most contagious. And naturally, as far as social distancing is concerned, we tend to stay away from people who really look ill. We don't embrace them, kiss them, hug them, and get close to them. We normally stay our distance when we're confronted with somebody who really looks ill. And this is the case for SARS coronavirus 1. is to say from 2002. The real problem with SARS coronavirus 2, the current epidemic, is that 30% of the infected individuals, they're asymptomatic. They show no signs of disease. And not only do they not tell the people around them that they are ill, many of these people themselves don't realize that they are carriers. And unfortunately for all of us, it is a proven fact at this point that asymptomatic, otherwise healthy individuals who have been infected by SARS coronavirus 2, they are infectious and they're spreading the disease. So whereas on the one hand, I've heard a lot of language about, well, the kids, they'll tolerate the disease, they don't even know it, they're not ill, they may be infected, but this is not serious. But if the kids who are not really seriously experiencing a difficult disease are nonetheless asymptomatic and infectious, every time they meet with other kids and meet with their parents and grandparents, they're spreading the disease. And so this is a very different epidemic. And whereas they both have very similar genetic composition, they have very different clinical behaviors. I think I've addressed your 3 questions.
Thank you. The questions are coming fast now.
Good.
One of the questions says, we never managed to find a vaccine for HIV. What are the chances that we will not succeed in having a vaccine for COVID?
Okay, that's a good question. I started my career, in 1980, a few years ago, working on rabies virus. And that I mentioned just historically because Pasteur was the developer of the rabies virus vaccine. And then I moved to the NIH and worked with Robert Gallo, who discovered the first human retrovirus, the second human retrovirus, and the third, which is HIV-1. And I started working on HIV/AIDS. In fact, I have been working on AIDS since 1987, 88. We still do not have a vaccine. And so this is a fact. I've had the honor in working with people like Mark Wainberg from Canada, as I mentioned previously. And I also served as the director of the Israel AIDS Task Force, our major NGO in AIDS. So I'm very intimately involved with the community and AIDS research. These two are very different viruses, HIV and SARS coronavirus 2. HIV is a retrovirus. It also has RNA, but for this particular virus to replicate, it has to go through a very unique process of generating a DNA copy of its RNA genes. Now, this is exactly the opposite of what I said when we started. I started saying that our genes are in DNA, and in order to generate proteins based on that information, we have to have an RNA copy. HIV has an RNA genome, genes, but it makes an intermediate DNA replica. And this process tends to be prone to mistakes, mutations. And so HIV is a very rapidly moving target. It's constantly changing its composition. And therefore, we have not been successful in devising an effective vaccine for HIV for a number of reasons. First of all, because HIV, as I said, is a rapidly moving target. It mutates aggressively because of its unique biology, biochemistry, the need to go from RNA to DNA and back to RNA. Another thing that HIV does is it targets exactly our immune system. And so every successful infection of our cells by HIV consequently reduces our ability to fight the beast because it's infecting our armory, our immune system. And so these two factors make it particularly difficult. And the third factor is is that HIV tends to hide within our cells and in our lymph nodes. And they're very large reservoirs of HIV, and we can't get rid of. Fortunately for us, the transmission of HIV is much, much less effective than that for COVID-19. Transmission of HIV means exchange of bodily fluids, either by blood transfusions or by sex. And so in some respect, if we assume very safe behavior, there's no reason why you should get infected by HIV. On the other hand, COVID-19, as I said, is an RNA virus. It's a very large RNA virus. In fact, it's a representative of the largest family of RNA viruses that we know. And because of this, it has evolved a unique system to ensure that when it replicates, it maintains the integrity of its own genes. So it's got a unique mechanism to repair its genes. And so RNA viruses that are stable, such as measles and polio, these are viruses that do not change and don't change, and therefore, if we generate a vaccine for them, the vaccine generated today is going to be very effective tomorrow and possibly next year as well. And so I'm very encouraged with the fact that I think we have very good chances of generating a vaccine for coronavirus, despite our experience and total lack of success for HIV/AIDS. And I'm certain that the effort that we and others are making in developing a vaccine today is going to prove very useful in maintaining and continuing to produce vaccines in the future. Now, it's I think important, if you will, I'll introduce also the question about flu, because we've talked about the extreme difficulty in HIV and the lack of success, the optimism and the success of generating very effective vaccines for stable RNA viruses like polio, measles, and I hope corona. But flu is intermediate. And we all experience that we have to get our seasonal flu shots. So first of all, I strongly recommend get your seasonal flu shots. That's important. We have an interesting phenomenon because we want to protect the elderly, or at least the older. I'm in the class of the older. And so the suggestion is that the older population should get flu shots. My experience and my professional suggestion is that one of the ways to ensure that the elderly, the older population is best protected is to start early. People who begin to take flu shots early on in their 20s, 30s, and 40s, and continue to the age of 60, of 20 years of flu shots from 40 to 60, that gives our body a lot of experience in dealing with the variations of flu strains. And so I think, again, that the flu vaccines are relatively effective. The problem there is that the virus is constantly changing, unlike coronavirus, and therefore flu vaccination is worthwhile. It's not as effective as polio and measles. And I would assume that it's not going to be as effective as the coronavirus vaccines, which I believe are going to be very effective.
Can I gather from what you just said that the likelihood of the mutation of the COVID virus is less likely because it's not going to change? Is
so
that?
I'll just repeat the question.
I think to deduct from what you just said.
Yeah. So Gladys is asking if we can assume that the coronavirus, the SARS coronavirus 2, despite the fact that it's going to change here and there, it's not going to be dramatic and it's going to stay stable. And in reality, there have been now hundreds of isolates of examples, samples of the COVID-19 virus taken from all over the world and use this material to actually map the diversity and the dynamics of its mutations. And so we can say, at least with confidence, that thus far, looking at the virus that we're confronted with now, it is not very dynamic. It's not changing rapidly, and it doesn't seem to be changing with any specific pressures to select unique and totally different virus. That doesn't seem to be the case at all. It looks like it is a very stable virus, and therefore, in that respect, a very good target for vaccine development.
Very good answer. And I have a few questions around how does the economical side of developing a vaccine agree or is compatible with the scientific? And is there a race by different countries, different companies, different scientific approaches to get the vaccine? Or will we be confronted with different vaccines and we will have our choice of to choose which vaccine we want?
Okay, well,
so
first,
is there going to be a lot of vaccines?
So I think that most likely, at least in the first round, the first generation, there are going to be a number of vaccines, because we know there are already a number of players that are investing huge amounts of money. And where people are investing huge amounts of money, they want to see a return on their investment. So even if, let's say, Johnson & Johnson makes it to the finish line before Oxford or others, I'm certain that you're going to see a diversity of vaccines. And I wouldn't be surprised that each company and each vaccine is going to tote that it is particularly effective and possibly even better than the competition. The reality is this: it's not enough to design the vaccine, as we are doing, and even to generate a prototype, but the manufacturing is going to be a huge, huge effort and hurdle, because we've never been in a situation where, in order to actually implement the product of our research at a level of having to supply 500 million or 1 billion doses to vaccinate the world in order to combat COVID-19. And if we want to generate a herd immunity, we don't want to do this over multiple years of vaccination, but we want to do it now. And so I think that what's going to happen is that a number of vaccines are going to hit the market. There are going to be multiple efforts in various countries and in various pharmaceutical companies to make as many as they can. And as many as they make, it's not going to be enough. And so therefore, we're going to have to use the products of different companies. As I said previously, I believe that the most important contribution that we may have is we may have a very effective, safe, and relatively cheap booster. And so again, I think most people are going to want to get their first vaccine. It may not be totally effective. We may see that it is marginally protective, but then they will want to get boosts. And the boost could be the primary vaccine or our vaccine or others. And so this is going to take a huge investment. I know there are a number of companies, Johnson & Johnson has made it very clear that they're taking the risk and they're already gearing up the industry, the production line, even though they do not have a proven and licensed vaccine, because the next step of churning out millions and millions and millions of doses is going to be a huge obstacle, and it has to be something that we're able to cope with. And this cannot be done overnight. So again, this is a long-term effort. Even if we get approval, the question is how do we get all the doses out there and distributed to the general public. And this is a major effort.
Thank you very much for your answer. We're coming very close to the end of this conversation, and we have a lot of questions around, and I'll turn it to you and say, Professor Gershone, how are you planning to, now that the lockdown is done with, will you live your life like the new normal? How will you live it? And what advice do you have for us?
Well, first of all, happily, because that's really important. But I'm concerned. I can't say that I'm totally encouraged, because if you walk around Tel Aviv and you go to some of the places and you see the rejoicing enthusiasts of one of our latest football teams, this is worrisome, because people have misguidedly and definitely erroneously assumed that we've passed the Corona crisis. Oh, Corona no longer exists. En kvar Corona. And so this is basically wrong. And so we have to educate. We have to explain in terms that the general public can understand that social distancing, donning masks, hand hygiene, these are easy tasks that need to be implemented as much as possible, and in doing so, allowing the economy. Now, I heard a very interesting little anecdote just before I came here. I was talking with one of my colleagues, and she has children in kindergarten. And we were talking about how was it with the kids when she was in lockdown. And she said it was a wonderful experience and so on. And now that the kids are back to kindergarten, she's once again confronted with head lice. Now, what does that mean? It's a very clear illustration that once the kids are meeting with other kids, they are not keeping social distance as you would expect. And fortunately, there isn't as much COVID-19 in kindergartens as there are head lice. But the mere fact that for the duration of the quarantine, she was free of head lice, the kids, I'm assuming that mom as well. And then once the kids are back to normal, we are once again confronted with contagious situations that involved closeness, close proximity, and interaction, which is proven by the fact that she's confronted with the natural situation in kindergartens around the world, is illustrated that we have to be concerned, we have to be aware, and we have to take care. And I think that with some degree of respect and understanding, this is quite feasible without totally locking down and knocking out our industries.
Thank you very much. We're coming to the end of our session. The 60 minutes are over. So do you have a final advice for us or a final word you'd like to leave us with? And after you've answered, I think Sharon Frankel will end the session. Answering our questions. Thank you, Amos, for letting this webinar come to the Spanish and Portuguese. Thank you, Consul General, and especially big thanks to Professor Gershone for taking the time and sharing your very deep research with us. Thank you.
So I would like to say just
Professor Gershone, last
just
word.
a few words of thanks. My father was a professor of the Technion, which is a fine school, by the way. And I'm a graduate of Hebrew University, which also is a fine school. And I also did some tour of duty at the Weizmann Institute in biophysics. And because of my experience about higher learning in Israel, I'm proud and chose to be a professor at Tel Aviv University, which is certainly a unique place, has supported our research, and has provided an atmosphere, multidisciplinary. We are the only campus that have all the disciplines on one campus. We are the largest university in Israel. And we have an amazing, amazing group of scientists and colleagues and teachers and faculty that are working together to try to deal with a very serious problem. And I'm delighted and honored to have had the chance to speak with you. And hopefully, maybe in the near future, we'll be able to talk about a new vaccine. Thank you very, very much.
So I would just like to continue
Thank you.
on your words. Thank you. I think I speak for everybody at Tel Aviv University, are honored that you are with us, one of our leading researchers, giving us the opportunity to even have and hold these kinds of sessions. I think that I speak for everyone, and I'm very confident that we definitely did not overpromise or underdeliver. I think that you have superseded all our expectations. We have another 20 plus questions, and with your permission, Professor, maybe I can send them to you over the next couple of days or so. If you have time, pick some to answer, and it would be great to get back to our viewers and supporters and donors with some answers. I don't want to put you on the spot, but I think it would be.
No, it's okay. I'd be happy to.
Wonderful. Thank you very much. I would like to thank everybody. Again, please support us. The campaign is on for another 3 weeks until June 30th, Tel Aviv University.crownchange.net. And you have heard what your support can lead to, what Professor Gershone and his colleagues are doing at Tel Aviv University. Thank you everybody for your time. Thank you for joining. And we hope to see you in one of the upcoming webinars very soon. Thank you, Amos. Thank you, Consul General.
Thank you very much.
Gladys, Professor, thank you very much.
אני אמסור דש לרוני.
להתראות לכולם. תודה שרון, תודה לכולם.
תודה לכולם.
תודה לכולם. בהצלחה.
תודה לרב, תודה לקונסול, תודה ג'וני, תודה גלדיס. ביי ביי.
ביי.